All right, today we're going to dive deep into the architectural blueprints hidden inside, well, every living cell. We're talking about these incredible microscopic structures that hold the master plan for all of life as we know it.
So here's the big question, right? What exactly are the blueprints for all living things? I mean, think about it. When you look at anything alive, from a tiny little virus all the way up to a giant whale, what is actually holding the instructions to build it? What is that master plan?
Well, it all comes down to these giant molecules called nucleic acids. Now, I know, that sounds kind of complex, but trust me, this is the fundamental chemical basis for pretty much every living thing we've ever found. It's the core of it all.
So, to really wrap our heads around how these massive molecules work, let's use a super simple analogy. Just picture a bead necklace. You got it? Okay, because that necklace is going to be our guide for the next few minutes.
Okay, let's start with the whole thing. The entire necklace, right? That long, continuous chain of beads, that's what scientists call a polymer. And it makes sense if you break down the word. Poly just means many, so a polymer is really just a long chain made of many repeating parts. Easy enough.
So, if the whole necklace is the polymer, then what about each individual bead? Well, that single, basic building block is called a monomer. Mono means one. So you've got lots of monomers, the beads, linking up to form one polymer, the necklace. See? Simple, right?
And this is where we bring it all back to genetics. In the real world, inside our cells, that single bead, our monomer, has a very specific name. It's called a nucleotide. And this little guy is the absolute essential building block of our genetic code.
But you know what? We can go even deeper. What if we could zoom in on just one of those beads? Just one single nucleotide? Is it just a solid little ball, or is it actually made of even smaller parts?
Turns out, it is. Every single nucleotide is actually a three-part structure. You've got a phosphate group, then you have a sugar, and finally, this really important part called a nitrogenous base. And that third piece, the base, that's the magic ingredient. It's the part that actually carries the unique genetic letter, like an A, T, C, or G. So every single one of our beads is built from these three little components.
And here's how it all comes together. When you link millions, literally millions of these three-part nucleotides together, one after another, well, it's just like stringing beads on a necklace. And what do? You get that long, amazing chain we started with, a polymer, specifically a nucleic acid polymer.
Now, in the world of biology, there are two main types of these genetic polymers, and I guarantee you've heard of them. They're the absolute rock stars of the molecular world. On one hand, you have DNA, or deoxyribonucleic acid, and on the other, you've got RNA, which is ribonucleic acid.
So what's the whole point of these long, complicated chains? Well, it's actually pretty simple, but also profound. Cells use these polymers to store and carry all the genetic information needed for life. Everything. How to develop, how to survive, how to reproduce, it's all in there. They are, quite literally, the code of life. And think about how efficient this structure is. It lets you pack an insane amount of information into a microscopic space. It's like a biological hard drive, but way, way smaller.
Okay, so let's do a quick recap. Our genetic polymers are like a bead necklace. Each individual bead is a monomer, which we call a nucleotide. The whole necklace is the polymer. That's our DNA or our RNA. And that incredibly elegant structure, that's how it stores all the information for life. Simple, but so powerful.
And that leaves us with one last really interesting question to think about. We've just talked about how perfectly this whole thing is built, with millions and millions of tiny pieces all in a row. But what happens if just one of those pieces, a single bead in that massive genetic necklace, gets changed, or maybe it gets damaged, or even just put in the wrong spot? What do you think the consequences are for the entire living thing?